Welcome to STEM NOLA. My name is Dr. Calvin Mackey, and I'm happy to have you all tuning in. As a little background, we founded STEM NOLA 7 years ago to expose and inspire and engage communities in science, technology, engineering, and mathematics. In the last 7 years, we've engaged over 50,000 K-12 students, over 12,000 families, and over 1,500 college students and STEM professionals. One thing that I'm proud to say is that we've put over $1 million in the hands of college students over the last 7 years serving as college interns with STEM NOLA, going out and engaging the community. So thank you for tuning in. Parents, thank you for believing in STEM NOLA and trusting us with your most prized possession, your child, your scholar. STEM NOLA believe that either we're training ourselves from the neck up or the neck down. In the 21st century, our children will only have one or three options. Either they're gonna take something, break something, and/or make something. And if we don't give them the skills and education to make something, like make a living, make a life, make a difference, it only leaves them with the two options that we see on the news every night, and that's to take and break. I don't want that for my two children, and I know you don't want that for yours, and that's why we're here today to expose our children to the possibilities of the 21st century. This is my friend. This is Alpha. Alpha is a humanoid. A humanoid is a robot that tries to act like a human. This humanoid has 9 servos and can do pretty much anything physically that a human can do. This is our competitor in the 21st century. Alpha can actually do push-ups. Alpha can even show off. And Alpha can even dance. And like any superhero, Alpha has her own music. This is the machine. And the automatic machine in the 21st century will displace many workers. In the aftermath of COVID, you have to believe that there are many companies out there saying never again will we allow a pandemic or something that happened in nature shut us down. So COVID has exacerbated the possibilities of machines displacing people. If you're competing against Alpha, you will not win. The only way we defeat Alpha is by developing our heart and developing our head, training ourselves from the neck up such that we can control the robots so that the robots will not control us. That's why we're here. That's why STEM NOLA exists. That's why your parents want to make sure that you all participate in events like this so you can know the possibilities of the 21st century. Again, I'm Dr. Calvin Mackey, and I want to say thank you for tuning in. Now let's get busy innovating, creating, and making with STEM NOLA. Thank you.
Thought we were starting for a second time there for a second. All right. Hi, guys. I am Holly Griffin, and I am a student at the University of New Orleans, and I've been working with STEM NOLA for about 2.5 years now, and I'm really excited because I know it's been a little while since we've been here, but we are back and we're moving forward. I know it's been a tough few weeks, but I'm excited to get started. So we have a guest speaker here to talk to us a little bit today about our topic. We're dealing with biology, DNA, and chromosomes, and we are excited about that. And we also want you to be aware to please take pictures of what you're going to do today. We're going to be doing some fun stuff with some edible gummy worms you probably have, making some DNA bracelets. So send those pictures to webinar@stemnola.com because we really want to see them, and you can be featured on next week or future weeks live sessions that we have. So we're going to move forward with our guest speaker, and that is Natalie Sarvar. So we are excited to have her. She's a medical student at Tulane, and she knows a lot about biology. She studies it. It's what she wants to do for her life. So we're going to go ahead and listen to what she has to say and learn some stuff. So see you in a bit.
All right. Thank you, Holly. Good afternoon, everyone. Let's see. Yep. After a hard couple of weeks, it is so nice to see people coming together for this live event. My name is Natalie, and I'm a second-year medical student at Tulane School of Medicine. In this intro talk, I will talk a bit about my experience with science and also emphasize the link between resilience and scientific discovery. I knew since I was in high school that I wanted to be a doctor. I was really interested in biology and nutrition and passionate about the idea that doctors could do more than just treat the sick. They could also keep their patients healthy. Medicine truly blends science and culture because the healing we provide is informed by scientific study. But the care we are able to give and the comfortable environment we need to create for our patients depends on our understanding, listening, and seeing them as people just like me who need a little help. I studied at Colorado College for undergraduate, where I majored in neuroscience and double minored in physics and chemistry. I really enjoy learning about the brain because, similar to how medicine connects science with caring for people, neuroscience connects how we think and feel with the exact biological changes causing those thoughts and feelings. I also did research with 3 different professors while I was an undergraduate. My first summer, I looked at natural competence in soil bacteria. I know there's a broad range of ages in this group, so I will try to explain the research in a way that y'all can understand. But no worries if it is a little beyond where you've been learning in your science class. We all go to school to learn, so you should not worry about not knowing right now, and it is always good to ask questions so you can learn. Going back to my research, natural competence is the ability of bacteria to take up genes from their environment and then integrate those genes from their environment into their DNA. It's a little like if I am a bacteria and I'm standing next to you and you are a bacteria, my hair could change to look a little bit like yours. It is really cool that bacteria are able to do this, but it can also be dangerous because we use antibiotics to kill bacteria when they are infecting. And sometimes the genes the bacteria can pick up can make the antibiotics not work as well, or even cause a person to need a different antibiotic, which requires even more research to develop. If we can better understand how the bacteria are getting these new genes, causing them to change, we can better respond with antibiotics that will kill these bacteria. My next summer of research, I looked at mating type switching in yeast. The yeast I was working with had 2 mating types, kind of like human sexes, alpha and beta. If a population or group of the yeast was all alpha, about half the population could spontaneously switch to beta, so the yeast could reproduce. This would be like if a town had only men and half of the population changed to women to be able to continue life in this town. It's pretty crazy what we can see doing biology. The first research I talked about was biophysics, and the second was microbiology, but they both related to genetics, which is a huge field of research in biology right now. You are going to learn a bit about how genes are replicated today, which is key information you would need to do any kind of research similar to the research that I have just talked about. My final research at Colorado College was with the psychology department, looking at how framing can influence the efficacy of bias trainings in education. Psychology looks at human behavior, so it's a bit different than biology, but understanding both is crucial to understanding research that is important for taking care of ourselves and contributing to society through research. Since finishing at Colorado College, I have been at Tulane School of Medicine, and despite the pandemic and recent hurricane, I have come to love this city and the resilience of the community. I texted all of the students I tutor for the ACT at Warren Easton after the hurricane, and one girl texted back that the challenges are just building her and her classmates up for greatness in the future. She was absolutely right. While stressful in the moment, hard times and setbacks that are out of your control can allow you to take time to think and learn for the future. This is very similar to how science progresses through trial and error. In many research studies, we do not get the result we expect, but rather than getting dejected, we just modify the experiment and try again. Science is imperfect, but it is fun, it can save lives, and it requires brilliant minds like your own to work hard. Scientific investigation, which involves doing labs like the one you will work on today, also allows you to develop skills that you can use outside the biology classroom or a career in the sciences. It is important to learn as much as you can during your kindergarten through high school years to give yourself opportunities for the future. I would love to see everyone here continue in the sciences through either research, medicine, or teaching. But even if you choose a different career path, the skills you develop through activities like those with STEM NOLA will help you be better prepared. Enjoy learning about mitosis and meiosis. Thanks for letting me speak for you, and I wish you all the best.
All right, thank you so much, Natalie. That was awesome and very informative and also very inspiring. I love that comment made by a student that all these challenges kind of help us build up for greatness. Sometimes it seems kind of tough, but I think we're going to move forward and we're going to learn and get stronger and stronger from it. So it'll be good. So I hope y'all are ready for the experiments or the activities that we have today. We got some fun stuff, some easy things, I think, to kind of help us step forward and get comfortable with this again. So this is our agenda. We're going to go over it really quick to show you what we're going to do today. And I hope y'all are ready, have your materials and your cameras ready and your palettes ready. So here we go. We are going to do some vocabulary terms. It's about 4 terms, and we're going to learn about those. And it's basically what we're touching on in the activities that we're going to be making. We have our objectives, the purpose why we're here. Why are we here? What are we learning? Why do we need to learn this? What's important about it? And then our activity questions. That's going to be 3 questions that you are going to get the answers right to, because we're going to do our activities, apply those vocabulary terms, and you are going to understand basically everything that we're talking about today. So it's going to be really fun. We're going to do a DNA bracelet, and then we're going to do gummy worm mitosis. So you just heard that word, like Natalie said, that's something we're going to learn a little bit about. And we're going to come back to those activity questions and get the answers for them. And we're going to have a good time. So I hope y'all are ready. We're going to go ahead and get started with those vocabulary terms. So let's learn these really quick. And you may have heard these words before, or you may never heard these words before. It just depends on the grade level or your experience. So we have DNA, we have cell, we have mitosis, and we have genome. So we're going to start with DNA, and that is a self-replicating material that is present in nearly all living organisms, and it's the carrier of genetic information. So it's the special component you have inside of you that gives you brown eyes, brown hair, or maybe it gives you blue eyes, or maybe it gives you freckles, or maybe it gives you attached earlobes. You have so many different traits about you. Normally they're the same kind as your parents, but you have your own special set of DNA. Nobody has DNA quite like you. Everyone has their own special sequence, or their own special DNA. And then we have cells. Cells are these microscopic, they're super tiny things inside of us, and we can't really see them, but they're always, always, always replicating. And they die out, and the new cells, we have billions of cells, not millions, but billions of cells inside of us, constantly making new cells all the time. There's cells in everything. So in every living thing. And we have mitosis, and that is basically the process of that cell division. So since we have new cells replicating all the time, we have mitosis, and it's this process that basically splits them and creates new cells all the time. Like right now, I'm walking, I'm moving around, I have cells that are replicating and making new cells inside of my body. So you do too. You have all kinds of cells inside of your body, moving, living, replicating. And then we have genome, and that's a complete set of genes or genetic material present in a cell or organism. So again, the complete set of genes or genetic material that is present in a cell or an organism. That's all our genetic information. It falls under the umbrella of a genome. Okay, so those are our 4 terms. So we're going to go ahead and get started with our objectives, and we're going to see why we're here, what we want to learn today, what's the goal. So you're going to understand what DNA is. You're going to understand what mitosis is, what a cell is, and what genomes are. And you're going to be able to apply to the project correctly. And then you're going to learn how to build a DNA bracelet, which is going to be really cute, and you can wear it, and you get to tell people it's a DNA sequence, which is pretty cool. So I hope you are ready to get started with that. So I'm going to go over the questions of the day. And if you want, you can go ahead and listen to those and get your materials ready too. So our questions of the day is, what is the name of the molecule that stores our genetic information? What is that called? We just talked about it, and we're going to go over it again in the project. So don't worry about that. Sorry about that. So what is the name of the process that causes our cells to replicate? So that is also one of our vocabulary terms, and you're going to get it during our project. Don't worry about that. And then after mitosis, how many daughter cells are there? So you might be thinking, daughter cells, what is that? But that is something we're going to talk about in our second activity. Okay, so hope y'all are ready to get started. We're going to get started with our DNA bracelet. So I'm actually kind of excited to make this one because I can wear it around town and show it off, and it's a DNA sequence. So let's make sure that you have all the materials that you need first. So that is going to be beads, it's going to be scissors, and it's going to be string. So our beads, you want to have 4 different color beads. So I hope that y'all do. And if you don't, it's okay. We can make it work as best as possible. But ideally, you would want to have 4 different color beads. So I have mine separated. I have red, blue, green, and yellow. And if you don't have these colors, don't worry. If you have pink, orange, purple, white, whatever, you can use that, whatever 4 colors you want. And if you have a variety of beads assorted, just try to group them together as best as possible. Just try to match the colors. Then we also are going to need some scissors because we're going to cut our string, and you need a string that you want to make a bracelet out of. So I have this kind of thick, almost yarn-like string, or you can use actually bracelet string. You may have that. I know my niece likes to make bracelets and stuff, so she has tons of this. You can even use, this is like a fishing material too. They have some like that. Just get creative and use what you can use. You can even use yarn if you want to, just anything that you have. And then you might want to get a ruler so you can actually measure out if you want to get really specific and measure out, because we're going to do like 10 inches. So if you want to get a ruler, go ahead and get that. And while you were getting your materials, I'm going to go ahead and talk about the science behind what we're doing. So a little background information, a little story while you're getting your materials is in 2003, scientists finished a really big project, and it was known as the Human Genome Project. This project allowed us to completely code the entire human genome, and that human genome is the entire DNA sequence of the human body. And if your bracelet, the bracelet that we're going to make today, if you basically were to take the entire human genome, it would be a super, super long bracelet, 30,000 genes in the human genome. That is pretty intense. We're not making a bracelet that long. We're going to make it like 10 inches, like I said. So, and instead, we used a very small portion of the genome, and we use that portion as a large role in determining your eye color. So biology is the study of life, and it's knowing the genetic sequence of organisms. It helps biologists to learn so much more about the organisms and all the living things inside of us and around us. So we're going to go ahead and get started. I'm going to give you a little more time to get your material, so I'll take it kind of slow because I have a few more pieces of information to introduce to you so we can get comfortable with this. So move that. I wrote a little list out, and these are 4 bases that are inside your DNA. So a DNA sequence it's a double helix. It's a weird structure that almost looks like a spiral. And inside of that, we have 4 bases, and these pair up together. So I have adenine, I have thymine, cytosine, and I have guanine. These are 4 bases that are inside of your DNA, and they pair up together. So we have kind of like best friends. They never leave each other's side. They always pair up. And let me see if I can kind of make a little picture for you and see if this will work out. So, oh, there we go. Even better. Oh, no, thank you. Thank you for that assistance. So, like I said, we have adenine, thymine, cytosine, and guanine. And I wrote my color because I decided what color I want basically for each specific base. So we can get to that in a little bit, but I have these 4 bases, and a DNA structure kind of looks like this. It's what I call a double helix, or what scientists calls, everyone calls it a double helix. And you have these strands that go across, and there's 2 bases on each strand. But the thing is, A and T, that is adenine and thymine, they always stay together. So when I have this, it's going to be A or T and A. And then cytosine and guanine, they always stay together too. They are best friends. They do not leave each other's side. It would be C or G and C. They always stay together, or it could be C and G. They never leave each other's side. So this may seem kind of like an overload of information, or this may seem like what's going on, but it'll kind of make a little more sense, and it'll be a little better to understand and grasp when we make our bracelets. So my drawing is not perfect. I am not going to be an art teacher. I'm going to be a 3rd grade teacher. We might do some crafts, but I'm not an artist, but the bracelet will help make it a little more sense. So like I said, we have 4 bases, and they pair up together, and that kind of helps us have different sequences. It's basically like helping us determine eye color. If I have freckles, like I said, if I have brown hair, if I have blue eyes, different things like that. So that they pair up together, and you are going to go ahead and assign a color to your bases. So like I said, we have 4, and I did adenine with yellow, I did thymine with red, and you do not have to follow this specific thing. Like I said, if you have pink, or if you have these colors, you have pink, orange, anything like that, you can go ahead and decide, just assign a color to the base. So you can make a little sheet like I made if you want to, and go ahead and write the different bases, and then you can assign the color that you want. And while you are doing that, I'm going to go ahead and cut my string. All right. Or I'll wait. I'll give you a second just so that y'all can do it. All right. And again, these are our 4 bases. So it's going to make sense when we make the bracelet. But all right. But again, assign your color. So write out the bases and then go ahead and write out the color. All right. So hopefully that makes sense. And we're going to go ahead and move forward with cutting the string. So in doing so, like I said, I'm probably going to make it 10 inches. I might make it a little more. My paper is about 11 inches, so I think I'll cut it the length of my paper. All right. So go ahead, like I said, get your string and get your scissors, and you're going to want to go ahead and cut it when you measure it out to what you want. I wouldn't go maybe more than like 12, 13 inches. This length is about fine. Let's see. All right, if that makes sense. And you're going to cut 2 strings just like that. Alrighty. You want them, it's okay if they're exactly not the same size, but you want them pretty close. Alrighty. And make sure you're using string that it'll be easy to get the beads on, because you don't want it to be too difficult. We want to make our bracelet with ease. So if it seems like it's too difficult, maybe see if you have a smaller string, something that's not as thick. It just depends on the beads that you have. So like I said, and something that might be helpful too is you can separate your beads by color. I put mine in cups to kind of help me out. And since, like I said, maybe I can do this too. These two always stick together, and cytosine and guanine, they always stick together. So since I have yellow and red, I'm going to keep my yellow and red together. And since I have blue and green, cytosine and guanine, I'm going to keep those together to kind of help me remember. All right. So hopefully y'all are ready to go. Let me give you a little bit of time. Separate everything that you need to. And I'm going to go ahead and get started with making the bracelet. So what you're going to do is you have your 2 strings, and just give yourself maybe about 1 inch of the way. It's okay if it's not perfect, but I'd say about like half your finger, or a little less than half your finger, like the top part. That's about 1 inch. Give yourself some space. So, and we're going to tie them together. We're going to make a little knot. So it's about this big. All right. And I'm going to tie them together. And if you don't know how to tie a knot yet, go ahead and maybe ask your parents or ask an adult, older brother or sister, see if they can help you tie a knot. Because I know it can be kind of difficult when it is a little tiny knot, and you're going to want to double knot it or triple knot it. Or you may know how to tie a knot, and you may be an expert already doing this much faster than I am. So, alrighty. So, well, I think a double knot is sufficient. So you're going to want to do that. So that is what we're going to have so far. Almost looks like the chromosomes that we're going to make later, but with extremely, extremely long legs. All right. You want to move it around. Shake it, give it a pull, make sure that it is nice and tight, that you got a good string. Oh gosh, mine's kind of shedding a little bit. Ew. But you want to make sure that it is super tight and that it's ready to go. So, all right. And when that's the case, what we're going to do is we're going to practice, or we're going to make a certain DNA sequence. So I have one. I'm going to turn this page over. Hopefully I have all the information. I can turn it back if need be. But this is a human sequence. So this may seem crazy, but these are our bases. And like I said, these bases go together and they make certain sequences that kind of help determine the color of our eyes. Like I said, if we have freckles, if we have a certain hair color, if our earlobes are attached to our face, different things like that. And so this is a DNA sequence for determining your eye color. So I'm going to go ahead and write this out, and it's going to seem super long, but it's just going to make us have a super beautiful bracelet. So I've got this combination of letters, and then I'm going to kind of test y'all on something too. All right. Towards the end, I had to squeeze my letters a little bit. So this is our sequence, and I know it seems crazy. I might have to rewrite it. So all right, make it a little clearer. And like I said, this is the DNA sequence for determining eye color in humans, because we're not the only people that have genes, and we're not the only people that have DNA. There's also animals and plants that have DNA as well. So this is our sequence. And like I said, I'm gonna see if you can remember, we're gonna make a pair. So I have one line for this specific sequence, but now I need to find the matching pair that's going to go on the opposite side. So we're going to put one bead here, and then we're going to put one bead here, but it needs to be that pair. So remember I said T and A are always together. That's thymine and adenine. And C and G, cytosine and guanine, they are always together. So I'm going to see if you can finish out this sequence. So remember, I'm going to show you A and T are always together and C and G are always together. So you're going to finish out the sequence. So I have a T, so I'm going to have an A. I have a G, so I'm going to have a C, A and T. So see if you can finish it out and see if you can maybe finish it out even before me. So here we go. There we go. So this, it seems interesting, but this is basically going to be our instructions for making a bracelet. So we're going to go with the different colors for this. So remember, like I said, you can even write it. I have this. I'm going to go over it again. I have A, mine myself. A is yellow. And T is red. G equals green. And C equals blue. So we're going to go ahead and make our bracelet. So it'll make sense too when I start putting the beads on. So my first letter, like I said, is T. So what I'm going to get is a red. So we may have different colors, like I said, but make sure your bracelet or your strings are nice and separated. And then you're going to kind of feed it through carefully all the way to the end on one string. And remember, it's a sequence. So T and A are always together, C and G. So since I put a T here, I'm going to go ahead and put a here, which is a yellow. All right, so you're going to go ahead and make your sequence, basically. And so I have C is blue, and I have G is green. So did anybody use any different colors by any chance? Or is anybody maybe using like purple, orange, or black? I don't know. So, and I really hope y'all take a picture of this too, because I would very much like to see what y'all's bracelets look like. And so I know it can be kind of tedious, so we're gonna have to focus in. So I put a green, and that's why I made this little code. So now I need a blue to partner up. And once you start doing it more, I think you'll start doing it faster. So I have T again is red. All right. And then I have A. So that is my yellow. Then I have G. And then that means that I'm going to have, goodness gracious, G in blue. All righty. And so, like I said, this is our DNA sequence. And everybody has different DNA. And so you actually have a really, really similar DNA to your mom or to your dad. You have it to your parents. But it's not quite the same. You have very different DNA from them in a sense, meaning that you don't look exactly like them for a reason. All right. And so A, that's yellow. And let me see if I can feed it through. I'm going to have to remember the sequence. So I don't know if y'all are having a tough time. Is it easy? Is it hard? Or maybe you're going way faster than me. Maybe I need to pick up the pace. So let me know, because I might be going slower than y'all. All right, let me see. So I stopped at A, so I need to move on to G and C. Yeah. All right, and that is a good idea. So I have got this done so far. So a good, maybe helpful tip is you can start crossing out stuff as you go. I've done all of these so far, so I can kind of remember, because this is a really, really long sequence. And like I said, it's determining your eye color. So I wonder if y'all have blue eyes, if y'all have green eyes. Some people in this world even have purple eyes. So very interesting. It's a very rare thing. I've never met anybody with purple eyes, but it is a thing nonetheless. All right, of course, I have brown eyes, as maybe y'all saw. All righty. And I haven't made a bracelet in a long, long time, but I kind of like this. This looks cool. I definitely want to see y'all's pictures because I want to see what colors y'all use. All righty. And let's see. It's a very tedious process, but the end result is really, really cool. And then when people ask you about your bracelet and why it has so many different colors, you can tell them that it's a DNA sequence for selecting your eye color. Alrighty, and keep this marker handy. So we're getting to that halfway mark. All right, and I'm excited about the end result. It'll look really cool when we are done. It just takes a little bit of time, but patience is a good thing. Something I think we all need more help with. Oh my gosh. All righty. Let's see if I can feed this through. All right, so I got TNA and I got CGTA. Gotta do it again. And it's okay if it's not perfect or you mess up, you can always go back and fix it. And if you don't, that is okay too. But I think we're gonna go ahead and get there. So T and Z, now I have A and T. So, see, I feel like I'm starting to get the swing of things, so maybe you are too, and maybe going a little bit faster. All right. And so with DNA too, people can actually test for your DNA and figure that out. And your DNA is located in so many different things on your body. So like your fingernails and your hair, different things like that. So if an eyelash falls on the ground or something, that contains your DNA in it. It's significant and it's special to you. Oh, my string is starting to unravel a bit. All right. Jeez Louise, I might need to cut this. There we go. And if your string's unraveling too, mine was kind of having difficulty with that, you can go ahead and maybe cut it. Might help you out a bit. All righty. And where was I? I should have been crossing out my stuff. I'm at C and G. I did that. I have 2 T's and then 2 A's. So remember, T and A, they always stick together. When you put a T, you're going to put an A. When you put a C, you're going to put a G. That's why we have our little chart. Because I need help remembering too. Oh my gosh. All right. Here we go. We're getting there. Don't worry, guys. Don't give up. We are getting there. We are almost there. Okay. And look at me. I think I did the wrong combination. Yes, I did. Okay. It's all right. I made a mistake and I fixed it. It's all good. All righty. And if y'all are already finished, goodness, I am getting there. I am so sorry if I am behind. I'm trying to finish this bracelet. Is anybody finished already? Or are y'all still working on it? Any chance? All right, now I have my C and I have my G. All righty. Moving along. I know this bracelet looks so random, but then when you tell people about it, you'll be like, no, it's a DNA sequence. Don't get it wrong. I didn't just mix up colors. This is a very strategic thing that we did. So here we go. We're nearing the end.
Jaden Moore said he's still working too. So.
All righty. Yeah, take your time if you're still working. Don't worry about that. Absolutely take your time because you want your bracelet to look really cool. Like I said, this is the DNA sequence for your eyes. So it'll be cool when you get that finished result. It's worth it to take your time. Don't worry about it. All righty. We are getting there. I thought I wouldn't see the end, but look at that. Wow. All right. My yellow. All right, let's see. And if your strands like me are kind of getting frayed, mine are kind of, I don't know if you can see it coming apart and separating. That's why we made that string really, really long too. I mean, you want to make it for the size of your wrist, so don't cut it too short. But if you need to, maybe cut a little bit off so it can kind of be straight in a single thread again. All right, I got 2 G's left. Oh, and look at me, grabbing the wrong thing. All righty. of blue, and I have one left after this. Hot dog. All right. I'm kind of excited about this now. And I don't know if any of y'all are professional bracelet makers where you have like those little metal hoops that you stick. I don't have anything fancy like that, but we will make do with what we have. It is innovation at home, so we're going to make it work. It is fine. All righty. Let's see. Last bead. Success. That is my DNA sequence. Okay. It looks crazy. It looks like a bunch of random colors, but it'd be really cool if we could put it in kind of that double helix like I showed y'all that shape. If you can kind of picture it. This is what it is, and they spiral. See if I can maybe get a little spiral. Something like that. Sure, we'll do an infinity sign for now. But they pair up together. So every time, remember, you have adenine, you have a thymine right on side of it, thymine. And every time you have cytosine, you have a guanine right on side of it too. But we have different combinations of these things. We have different sequences all in our body because we all have different DNA, and we all have our own special fingerprint. I know you probably can't really see it too well, but you all have your own special fingerprint. Nobody has DNA quite like you. So they can tell who you are by your hair, by your fingernail, like I said, or by different things if a skin flake comes off, because we shed skin all the time and we shed hair all the time. So that has your DNA inside of it. So now hopefully you'll have that complete bracelet. And if you're still working, don't worry about that because I'm not quite finished yet, but I'm getting there. So we got all of our beads on, but now to complete this bracelet, make sure these beads don't fall off. Remember how we did that double knot? You may want to do a triple knot to make sure it's just really secure and it's a big knot and the beads don't fall off. You're going to go ahead and tie a knot as close as you can to those beads right here so that it doesn't separate and it doesn't fall off. So I'm going to go ahead and I'm going to try to triple knot it. So be careful. I'm going to kind of bend down and do it too. Let me see. All right. So, and again, if you need an adult to help you, maybe they can help triple knot it, even quadruple knot it. That means 4 knots. I think I'm going to go for the 4 knots because I can do it. And you can too. Just takes a little practice. All righty. Let's make sure. And I'm going to go back and just kind of make sure this is nice and snug because I want to wear my bracelet. It is a nice fashion statement, I feel. Okay. And I hope it fits. It fits. Okay. Oh, wait, can you see it? I like this. It was worth that hard work. I almost wanted to give up, but I didn't do it. No, there's a lot of beads. So I kind of like this bracelet. I'm not going to tie it just yet because it might take a little bit of time. But again, if you, like I said, a professional bracelet maker, like my niece is, you can add these little metal hooks. You can get them at like, an art store or something and attach them on. It has a little clip and then a little metal hook, and then you can just kind of attach it. Or you can even add it to a necklace. Maybe make it a headband. I don't know. Do what you want. You can stick it in your hair. I'm going to make it a bracelet. I like a bracelet. So if anybody's still working on it, don't worry. Take your time. I'm just going to put mine to the side. All right. So we can go ahead and move forward with our next activity. So we just talked about DNA. Oh, done. Awesome. So I'm sure you have a great bracelet. Be sure to take a picture of it. I want to see it. We all want to see it. All right. So like I said, we just talked about DNA, and now we're going to talk about mitosis. So we're going to go ahead and get out those gummy worms that you probably have. You may have gummy worms. You may not have gummy worms, but that is okay. Where is my paper? All right. Going to go ahead and get ready. So this is our mitosis activity with our gummy worms. It is dealing with chromosomes and DNA, and we're going to talk about that process. So I want to make sure you have the right materials. Again, not too many materials, but we're going to go ahead and go over that, and then I'll go over the science behind it. So materials, you need gummy worms, you need construction paper, and it, again, doesn't have to be construction paper. I'm using something called cardstock or bright paper, so it's kind of thick. You can use you want something with a plain background. It could be poster board, it could be a napkin, just whatever you got, use that. And you just want it to basically be a plain background, no words, no writing on it. You just want something basically like this. And then a marker. So I have a Sharpie, or you can use colorful markers, but just make sure that you can read it. If you have a yellow paper, you don't want to use a yellow marker. And if you don't have gummy worms, maybe use something like Twizzlers. You can use just any kind of candy that has that line structure on it. And if you don't have candy, you can use pipe cleaners and you can bend those. So just get kind of creative, or if you want, even you can use your string if you need to. So if you don't have gummy worms, don't worry. There's tons of things we can use for this. So I'm going to go over the science, like I said, while you get your materials and get ready for this. And if you're using gummy worms, maybe wash your hands. So if your parents say it's okay, maybe you can eat them later. But eventually, cells need to duplicate. And this is for mitosis. So our cells, like I said, we're constantly reproducing cells with these little baby microscopic things inside of us. It's constantly reproducing all the time inside of us. It's cells, and they need to duplicate. So there's two main forms of replication. We have mitosis and meiosis, but we're going to go over mitosis today. And the tutorial talks about mitosis. It's a big idea to remember that mitosis is just a simple duplication of a cell and its parts. So you have something where DNA duplicates and you have two new cells, and that's called daughter cells. You have one parent cell, it splits, and you get two new daughter cells, and then go together, split. You just have this ongoing process that never stops all the time. And it's two identical copies that come from one original. So it's basically like I took this piece of paper, split it in half, something like that, and it's going to keep replicating. And it occurs in 4 steps. And these steps are all a part of mitosis. Mitosis is the process of it. And we have something called prophase, we have metaphase, anaphase, and telophase. That's 4 different processes that happen. It's different steps in mitosis. And it's the separation of the cells and then getting together. So we're going to go over that. So I hope you have all your materials, and we're going to go ahead and get started. So I've got my gummy worms, my little baby gummy worms. And again, it's okay if you don't. Use whatever you need. Like I said, you can use string, you can use pipe cleaner, Twizzlers, anything like that. So we're going to kind of draw this out. So get your piece of paper ready, get your marker ready, and your gummy worms, and let's get started. Okay, so we have our first thing. We're going to draw a nice size circle, and we're going to label it prophase. Can you say prophase?
Prophase.
I'm sure you said prophase. My coworkers here have said prophase. They did a great job at pronouncing it. That's our first stage of what we call our cell division. What is this process called? It is mitosis. My gosh. All righty. So basically, I need to make sure my gummy, if you have gummy worms and they're split, different colors, maybe try to make sure the colors match with one another. If they don't, it's okay. It's not the worst thing in the world. So basically, you have these things called chromatids. It sounds crazy. You have chromatids. And it was once a chromosome, and it's separate. It's chromatids. And they separate from each other, basically duplicating, making a copy of itself like we just talked about. So this is the first stage. Chromatids, they've duplicated, and now we have 2 individual chromatids just waiting to move forward on that next stage. Chromatids, I pronounced that so wrong, and I just got informed that I pronounced it wrong. So we all make mistakes and pronounce things very wrong. So please don't say that it's chromatids. I'm so sorry about that, guys. All righty. You're going to draw your next circle, and that is going to be your metaphase. All right, and what happens with this is we have these things that kind of come about, and they're called spindle fibers. All right, and then all of a sudden, from your duplication process, you have your daughter cells. So they're together, and now these are chromosomes. So an individual one, remember, it's not the way I originally said it. It is a chromatid. And so that is the way you pronounce it. But when they come together, they connect in the center. So it may be kind of tough to see, but they connect in the center right here. And they have these legs, it looks like, but they connect in the center, and that's called a chromosome. A chromosome is a pair of 2 chromatids. All righty, and that's our metaphase. So now that we went from this, our duplicate process, and we went to metaphase. So I'm going to go ahead and draw another arrow, and yours doesn't have to look exactly like mine. I just feel like this is the way it makes sense to me. When I talk about a process, I like a good flow chart. All righty. Draw a nice, good circle. And we're going to label this anaphase. Can you say anaphase? I'm sure you did. I heard my people say anaphase. Did a great job at it. So anaphase now, we just got these beautiful little chromosomes, but now, so sadly, not sadly, it's part of the process, but basically they were once together and now they're going to separate from each other. Those spindle fibers that are on the side, they're attached to these chromatids. And they kind of hold on to them and pull them apart. They say, we're going to separate you because we're going to make something really cool later, but we're going to separate you, and they go to opposite ends of the pole. So like on the Earth, we have a North Pole and we have a South Pole. So if the Earth was like this, but it's okay. So they go to the poles and they separate from each other, and that's anaphase. They move apart, do this separation. All right. And like I said, it's kind of almost looking like our prophase in a sense, but we have two copies, we have two daughter cells, and they separate from each other. And then we're going to go to our last phase. So make a nice big old circle for this. And what this phase is called is telophase. So can you say telophase? I'm sure you can. They didn't want to say it this time, but I'm sure you can say it. So it's all right. So what happens now is you've got your duplicate process. You have brand new daughter cells. So you just went through this long process of cell division. Like I said, it constantly divides all the time. Your cells, you have one and it's constantly replicating. Those old cells inside of you are dying and those new cells are coming to life. It has to happen all the time. And this is mitosis. We have prophase. Remember, metaphase is our second. And then we have anaphase and we have telophase. These are these chromosomes and these DNA. It's inside of our cells. It's inside of something called the nucleus. And the nucleus is like the center of the cell. It's like the brain, basically. And it's got the chromosomes inside of it. And inside these chromosomes right here is DNA. It's really, really tiny. You cannot see it with your eye, but this happens all the time. It's constantly splitting and then coming together, constantly splitting and coming together. So this is our process of mitosis. So this isn't something we can keep, but you can take a picture of it if you want to. And then you can also maybe take a little treat if your parents say it's okay. So that is our cell process. This one was pretty easy. It's more of a demonstration. Like I said, now you have your cool DNA bracelet. And like I said, this DNA is deep inside of your chromosomes, which are inside of your cells, which is all a part of genomes, all connected. So I feel like we did a pretty good job today. Like we had our bracelet and we had our little edible fun activity. So we're going to come back to those questions and we're going to see what you remember and what you learned. So our first question is, what is the name of the molecule that stores our genetic information? Remember, it's special to us. Our hair has it, our fingernails have it. Basically, it's all over our body. It's special and significant to us. All right, nice job. Yes, it is DNA. That is it. That is what we talked about. So now, what is the name of the process that causes those cells to replicate? They split, they duplicate, they come together, they split and duplicate. What is that process called? It's got 4 different stages in it. So we just talked about it. There we go. It is mitosis. Thank you all so much. Doing great, doing great. All right, and last one, after mitosis, how many daughter cells are there? So with this one, I don't think I specified it, but we'll see. Just take your best guess. Don't worry about it. Don't be afraid to get it wrong. We've all gotten things wrong, but I think you can maybe get it right. So how many daughter cells? If you have one parent cell and it splits, so I can see why you thought it was 4, but I think it's my fault. I didn't specify. When you have one parent cell, it splits and you get 2 daughter cells. So like I said, you have one parent cell and then it splits and you get 2 daughter cells. So I'm sorry if I didn't make that clear, but don't worry about it. We all get stuff wrong, but that's how you learn is you make mistakes and you learn. So it is all good. But I think that we did a pretty good job today. It was nice and easy, refreshing, I think, and a good way to come back. You get a nice, cool little bracelet or a necklace if you want. And so I think we had a pretty fun time, learned some stuff from Miss Natalie. And we want you to go ahead and tune in next week. We're going to be doing levies. So I think that's very pertinent and important to what kind of has been happening lately. Levies is a big part of New Orleans and a big part of Louisiana. So tune in next week and we will have really fun with that. And remember, send those pictures to webinar@stemnola.com. And I hope y'all had fun today. I hope y'all having a good time and easing back into coming back to the city. I know it's been a tough time, but I think that it's going to be good. We're going to have a good year and a good finish. So I hope y'all had fun. Thank you so much. And again, my name is Holly and I had a good time. I wish we could be in person, but it's all good. We had fun. So have a good day and a good week. See you next week.